Hi, I'm John Strum, and this past fall, I attended ECTRIMS, the largest MS research conference in the world. During the conference, I had some quick conversations with some of the experts who were on hand to present their work to the more than 9,000 attendees. In a moment, we're going to see one of those conversations. And then you'll hear from some folks who are living with MS who are going to share their perspectives on the topics we discuss. I'm at ECTRIMS 2023 in Milan, and I'm speaking with Dr. Jiwon Oh. Dr. Oh is a neurologist, scientist, and the medical director of the Barlow Multiple Sclerosis Program at St. Michael's Hospital in Ontario, Canada. And although we've spoken before, it's especially nice to be here with you in person.
It is, and thank you for having me, John.
Well, I know you're quite busy at this conference. You're giving at least 3 presentations that I know about, and your work on the extension study of a potential new disease-modifying therapy is being presented at the poster session. So I'm hoping you can explain what an extension study is, and then tell us what you learned in the extension study for Tolebrutinib.
Sure. So an extension study is usually after the initial phase of a clinical trial. So in a clinical trial of MS or many other disease states, people are usually enrolled and they're randomized to either the experimental drug or placebo or an active control. And people are followed very closely for a pre-specified duration. And after that initial double-blind period, if people are willing, they will enter into an extension study where most people will be on the experimental molecule. And the point of an extension study is for a longer period, you're able to monitor people to see if the clinical efficacy that was seen in the original double-blind period of the clinical trial is maintained, and importantly, to see if there are any new safety signals. And so for Tolebrutinib, in a phase 2b trial that took place over a period of 16 weeks, there was evidence of clear efficacy of the highest dose of Tolebrutinib at 60 milligrams that was used on the primary endpoint, which was the formation of new gadolinium-enhancing lesions. And so after that initial double-blind period where people were actually randomized to many different doses of Tolebrutinib, and there was a placebo run-in and run-out period, people who were willing were invited to enter the extension phase. And during the extension phase, people initially in Part A were on the same dose of Tolebrutinib they were on in the double-blind period. But then at a certain point, the company made the decision that everybody would continue on the highest dose. And so that's what people have been on, 60 milligrams for now 3 years. And so what we've been monitoring is how people are doing clinically, and importantly, if there are any new safety signals. And the good news is up to the 3-year mark, it seems like the clinical efficacy pieces were maintained in that people continue to have a low annualized relapse rate in comparison to the year prior to enrolling in the study. With respect to MRI measures of disease activity, MRI measures of disease activity remain low during the extension period. In terms of disability scores remain stable throughout the 3-year extension period. And then with respect to safety, there were no new safety concerns that have been identified to date. So this is all good news.
It is good news. You know, with over 20 approved disease-modifying therapies for treating MS that are available today, why are clinicians excited about this potential new category of DMTs?
So Tolebrutinib falls into a category of drug called the Bruton's tyrosine kinase, or BTK inhibitors. And there's many different BTK inhibitors being tested right now. So it's a really interesting time in MS because I don't think there's ever been a time in the field when simultaneously there's so many different drugs that belong to the same family are being tested. So lots of excitement, as you said. And the reason is because we know that BTKIs, at least theoretically, can have a beneficial effect on B cells in MS, which we know based on the existence of therapies that target B cells are a very important player in MS pathophysiology. But the reason why there's added excitement, because right now we do have many treatments that can profoundly impact B cells and are really efficacious for at least the relapse biology in MS. But with BTKIs, the reason there's so much excitement is because many of them can get into the brain. And at least in animal studies and a lot of in vitro studies, there's evidence that these BTK inhibitors affect a type of cell in the brain called microglia. And the reason we focus so much on microglia is because we know that this is a major driver of non-relapse disease biology, or CNS compartmentalized disease biology, which we think is responsible for the slow progression that we see in many people living with MS. So the hope is that not only do BTKIs have a very good effect on B cells, but that they get into the brain and beneficially modulate microglia, which we think are a major driver of progressive disease biology. And so it would really meet an unmet need in the field clinically if they do actually have an effect based on clinical trial data.
You're giving a presentation on the future of MS care, specifically how biomarkers might be used in clinical practice. So can you share some of your thoughts on how biomarkers might impact someone's clinical experience?
Yes. So, in any field, I think it's important to first note the immense progress that we've made because, I think in MS, it's an illustration of a chronic neurological disease where in just 2 decades, we've probably, increased the number of treatments that we have by tenfold. And so that is profound. However, there's a lot of things that we recognize we need improvement on. And one of them is better ways to monitor people with MS. And what I mean by that is, as people with MS will know, when they go into their neurologist's office, we have our old tools, the reflex hammer and the tuning fork, and we do our best to examine people neurologically, but it's so clear that the neurological exam is flawed, and we need better tools clinically to try to evaluate how someone is doing with respect to their disease activity. And again, we're lucky because we do have MRIs, but it's clear that the clinical MRIs that we use now are not really capturing everything that we need to. So this is where better biomarkers of monitoring disease may have an important role in the future. And there's lots of potential candidates, including even smartphone apps, digital tools. There's blood tests that may help us better monitor people's disease activity. And then there's some new MRI measures that may help us in the near future. So this is where there's a huge unmet clinical need. And this is where I think there's a lot of hope for biomarkers in the field.
You're also leading a presentation on approaching precision neurology. Can you help us understand what precision medicine is and how precision neurology may benefit someone who's living with MS?
So, I think in a disease like MS, there's so much variability in how individual people will present, what symptoms they'll develop over time, and how their disease will evolve over time. And, as clinicians, we do our best to try to prognosticate and predict in an individual patient in front of us, how someone will do and therefore what therapy would be best for them. Because again, in MS, we're very lucky because we have nearly over 25 treatments available. However, to be honest, we're not that good at doing that. And so this is what precision medicine is. It's taking into account many different things about an individual patient's disease state and making the best possible decision with respect to treatment options so that you can optimize clinical outcomes, not just in a year or 2 years, but 20, 30 years down the road. So that's I think it's not just in MS, but any chronic disease state. We need to do better at administering precision medicine, which means you need to understand the variability of a disease and make the optimal decisions that will be best for an individual patient. And this is where biomarkers may be able to help us in the future. And we're doing our best to do a good job at this, but there's a lot of room for improvement.
Well, Dr. Jiwon Oh, thank you for all you do to improve the lives of people who are living with MS. It's always great to talk with you. It's especially nice to be here in Milan with you. I'm John Strum, and this ECTRIMS Extra was made possible through a grant from Sanofi. We've just watched my conversation with Dr. Jiwon Oh, and joining me to provide some perspective from people who are living with MS are Rich Lavasser and Kara Skarupa. Rich, Kara, welcome.
Thank you.
Kara, when were you diagnosed with MS?
I'm coming up on my 13th anniversary of being diagnosed with MS. So Geez, don't make me do math. 2011, in the spring, early spring, I was diagnosed.
You just heard Dr. O share some encouraging results from the extension study for Tolebrutinib, an investigational disease modifying therapy that's part of a new category of DMTs called BTK inhibitors. What are your initial thoughts about what you just watched?
I was just super excited to hear where our drug, trials are going and how they're going, and the fact that there's this whole new class of drugs that looks to be incredibly promising. It's just exciting for me. I feel like, we as people living with MS, somebody like myself, there's there's so many differences in how the disease presents and how we live and how it affects us, that I think having a multitude of ways to attack the disease itself is incredibly helpful.
Rich, can you tell us when you were diagnosed with MS?
January of 2002, so 22 years ago.
Dr. O talked about clinical trial results, and I'm curious, have you ever participated in a clinical trial?
Of a drug, no. No, not a clinical trial of a drug.
Clinical research is how we get to next generation, next level medications like the one that Dr. O was just describing. Given the potential upside, why do you think recruiting clinical trial participants can be so challenging?
I'm not really sure why it's so challenging. I jump at the opportunity for clinical trials, always looking for ones to do. Um, I think people have a fear, uh, an intrinsic fear from the get-go of, okay, what if I'm put in the placebo category and I get nothing? Um, so there's a fear there. But I think with the things that she was talking about would certainly make this more uh, make people want to participate more than they do now.
Tara, do you have any suggestions for how clinical trial recruitment could be improved?
I'll echo that exact sentiment. I think a lot of it is fear based. I think if we had more of a relationship with individual neurologists in individual communities and included them more, excuse me, sorry, with the recruiting, it would be helpful because you would have a real person, with whom you have a relationship already telling you this is good and this is important. The other thing is that I think it would give us a better representative group, because demographic wise, we don't always get the people in the trials that really represent what our community looks like.
I'm curious, over the past 13 years that you've been seeing a neurologist, have you learned about any clinical trials?
No, actually, I haven't. I am involved in the MS community in a few different ways, and I'm actually involved in a trial now that it's not a trial per se, it's a study on the effects of the COVID vaccine among MSers. And I found that study myself, and I signed myself up for that study. I wish it didn't have to be so individualized. I wish there were something almost like a clearinghouse where people could go and see if there were a clinical trial that they fit the parameters of or not, instead of just waiting, for a lightning bolt out of nowhere to tell us that this is happening.
During my conversation with Dr. O, she also talked about precision medicine. Kara, what's your reaction to treating people with MS using this precision neurology approach?
Thrilled. I think it's long overdue. They've been doing it in the cancer community for a very long time with very good results. And as I said, we're kind of known as having a snowflake disease in that it presents differently in different people. So we have little quirky parts to us that, maybe doesn't exist in other diseases, but with MS, it certainly does. So to know that my MS is different than your MS is different than somebody else's MS, and that there could be something special about my MS that requires a different sort of treatment. So I think it's incredibly exciting to know that it will be more precision based instead of sort of a shotgun approach.
Rich, how about you? Any thoughts on how precision medicine might benefit people with MS?
Oh, when she said precision neurology, I get so excited because I know it. I, s- I see it happening with cancer patients, and it's targeted to the individual. If we could do this with MS, people with MS, it'd be a game changer. But you do have uh, the snowflake disease, like everyone with MS presents differently. So it's going to be I suspect difficult, but I'm looking forward to it. I was so excited to hear that. Using biomarkers, maybe that'll help target the precision neurology. It could be a game changer.
Kara Skarupa and Rich Levasser, thanks for spending a few minutes talking with me today.
You're
Thank you
welcome.
for having me.
Thanks for watching. And if you'd like to hear more about the amazing MS research that's taking place in laboratories all around the world, I hope you'll join me every week for the Real Talk MS podcast.